Well, thank you, Jan, for inviting me to give this talk. I really appreciate it, and thank you, everyone, for being here. And hello to familiar faces and names, and hello to unfamiliar faces and names as well. So, I want to talk about some work we did a couple of years ago, more than a couple of years ago now, which is about testing for significant changes in gene or bacterial population compositions from a small number of samples. And we decided to then, in the end, call this tool that we wrote DirtyGenes, which is a kind of a little bit of a spin on some of the next generation sequence analysis software names that but we went for something that was in the opposite end of the clothing spectrum. So this is the sort of data that we were looking at. We actually talked to Yongguan Zhu in Chinese Academy of Sciences, who some of you might know, and he had some data that is very typical of AMR data, particularly from the environment, where one might see abundance of antibiotic resistance genes or abundance of bacteria, and these will be presented in papers, and much more often than not, these will be presented without any kind of statistical analysis that might say whether or not the differences observed between the environments are statistically significant or not. And there are a number of reasons why that seems to occur, probably because it's difficult, but probably because also it's just not seems to be culturally embedded. So this was some example. It was just one of the data sets we used as an example. In this case, it was different amendments to some soils to see whether or not this had an impact on again, either antibiotic resistant genes by class or bacterial phyla. See a spelling error in the word bacteria there, which I haven't spotted before. Okay, so apologies for that. So at the time, we had some other collaborators over in our vet schools, such as Adam Blanchard and Piers Davis and Sabina Turtemeyer, and they were doing something similar. They had some data on sheep hooves. They got a very small project. I don't know if you remember, there was this EPSRC Bridging the Gaps award in the UK for those who were in the UK, and within Nottingham, this gave out lots and lots of very small projects, and again, this was measurements on fairly small number of sheep hooves using shotgun metagenomics, and they were again interested in knowing whether or not there may or may not be differences in antibiotic resistance gene abundances in different seasons, but based on what was really quite a small sample. And one thing, as you'll note, is that in the previous slide, we had some data from qPCR arrays, we had some data from 16S metagenomics, and here we have some data from shotgun metagenomics. So the data is actually coming from some quite different methodologies and different and approaches. And here again, okay, so we have the same here again, and this is now looking at the overall bacterial communities. So again, it's the same questions really, and one of the questions we wanted to know, or this group of collaborators wanted to know, is are these resistance genes or bacterial communities, have these compositions changed between the seasons, but again had a relatively small number of samples. So this is kind of where we got to, which was, you know, can we develop a statistical test to determine if there is a significant change in composition? This is questions like AMR genes or bacterial taxa. We want to be able to do analysis on the full data set. We recognize these come from a range of technologies. And can we apply the test method also for experimental design to calculate sample sizes? At the time, our collaborators, again, this is Sabina, were putting in a research grant proposal, and they wanted to be able to rationally decide how many, in this case, animals they put into the grant. And because it was involving sheep, they did have to have a kind of a formal experimental design methodology in there to justify how many animals they were going to use in the experiment. So we wanted to do that, and we were aware of some existing methods, particularly based on the Permanova method, but we were kind of the view that we wanted to do something that was a little bit different, that would be broadly applicable, but also applicable when we had actually some quite small numbers of samples, which is an important part of what we were doing. So I was really glad that Jan introduced Poisson distributions and even binomial distributions, because the starting point would be to say that it's something along these lines, right? So you might have some different colored balls in a bag, and you're trying to draw out blue, yellow, and red balls. And this is a bit like what you might think as being your sequencing. Let's suppose you're doing some kind of shotgun metagenomics for, as an example, where you might be doing something else. Okay, and you're kind of drawing out sequences and sequencing them. And so you might end up getting a random sample where, as you start drawing out these different colored balls, which might represent different sequences, you end up with some different probabilities, and they might be representative of the overall frequency of those genes or taxa or whatever it is you're interested in that community. But there's only one problem here, and it goes back to what Jan was talking about with these Poisson distributions, which is that kind of assumes that these events that we observe are indeed independent of each other. That if I sequence a gene and I sequence another instance of that gene or whatever it is, that those then truly reflect the underlying probabilities associated with the frequencies of those in the overall population. And the problem is that we know that this is not true, right? So we know that different technologies and different sequencing technologies or qPCR technologies or any other technology you might use all have biases. All those biases are different, and those biases might manifest differently if you're working with different samples or environments. So to make this assumption isn't quite correct. So what we wanted to do was therefore derive a general statistic that would allow us to check for differences between samples that would respect the fact that there may be non-independence in our samples, but would also do something very useful, which is called to be statistical literature is called universally most powerful, and that basically means we'll give you the most amount of information given the data you have available to you, which you can do with parametric statistics, and specifically you can do it with parametric statistics if you base your statistical test on what is known as a generalized likelihood ratio statistic. And what happens to the likelihood ratio statistic is as follows, all right? So if I've got, let's just say, two groups of samples from two different environments, but we can apply this to many groups of samples, all right? We've basically got, as you may be familiar from standard statistics, sort of two hypotheses. You have your null hypothesis, which basically says that there is no difference in the population structure between our sets of samples, and therefore all the variability we observe is random. It's all due to the sort of sampling errors that again Jan has mentioned. The alternative is to say, well, actually, there is a difference in the population structure, and therefore the variability we observe between our two sets of samples from, let's say, two different environmental conditions is in fact due to those environmental conditions. So there may be some internal variability within the environmental conditions, or as are associated with the sampling or the measurement technology, whatever it is. But actually, there is a genuine difference between the population structures. And the reality is, of course, that we've got a mixture between the two in any given situation, right? You've got some amount of random variability and some amount of variability that may be due to the different conditions. And the likelihood ratio statistic kind of puts an arrow into where you are between these two places, and allows you to accept or reject the null hypothesis in a sort of in a standard way. So this is what we did, and I'm not going to share with you the details of the mathematics. You can read that in our paper if you wish, but the basic idea behind it is that we work out this likelihood ratio statistic from our real data. And what we do is we don't use a Poisson distribution. We use something else called a Dirichlet distribution. And what the Dirichlet distribution is it's a bit like the multinomial distribution, which is the distribution you want to use, that assumes the independence of the of our draws. But there's another parameter that's involved that takes into account the fact that the draws are not independent. So it takes into account the fact that maybe within whatever technology it is, whether it's a qPCR technology, whether it's a particular platform for sequencing, there may be, once you start to get certain events, you start getting more of those events. And so they become more common than predicted, or indeed vice versa. Perhaps more frequent events are for some reason less well observed. There is a parameter, an additional parameter to be estimated from the data that allows us to take that into account. And so we use this Dirichlet distribution to do that. And the Dirichlet distribution is why it's called DirtyGenes, you see. That's the dirty bit, is this Dirichlet test. And so by having this additional parameter, we can take into account, at least to some degree, the bias introduced by the technology that does the measurement. Having done that, we generate a statistic from the real data. Again, as I said, the actual formula is in the paper if you want to read it. And then what we do is we create randomized data based upon the null hypothesis. Okay, and then we work out a statistic for each of those. And this is a fairly standard approach. What we do is if we assume the null hypothesis is true, you can generate data that might be like your experimental data under the null hypothesis. And then you can compare the distribution of your test statistic from your null hypothesis against your statistic from your real data. Basically, if you're in the belly of the distribution, then you're going to say, well, you can't tell your data apart from your null hypothesis, which means that there is no evidence to reject the null hypothesis, and the variation that you may observe between your groups of samples appears to be random, or you can't tell if it's not random, or if it's towards the edge of this distribution, then what we're saying is, well, look, it doesn't look like the null hypothesis explains this variation well, and so we would reject the null hypothesis as we would do with a standard statistical test and say, okay, well, it looks like the variation between our sets of samples appears to be something that can't be explained by random chance alone, and therefore there may be a difference between our environments. So I mean, just to say again, I said I've left out the details of the maths, but we did apply this in our paper to the samples that we had. So the first thing was we had the antimicrobial resistant genes in the foot rot, and I think now you have to correct me if I'm wrong because I haven't looked this up. And it's got, you know, as you can see, it's a few since we published the paper. I think it was about 4 sheep. So it was a really very small number of sheep that were in the experiment because that was the limit of the budget. But we still got a p value of sort of 0.087. So we had to kind of reject the null hypothesis, but we were kind of aware that the sample size was very low. So the power of the test was low. Whereas the bacterial taxa had a kind of a p-value of 0.822, so it didn't look like the community was changing a bit. Again, small number of sheep. The sewage sludge data that we had from the soil amendment from Yongwan Zhu's lab, so those came out to be highly significant, and again there was clearly enough data there to address that. So that was kind of what we did with it at the time. But then the other thing we did, as I said, we had 4 sheep. I think we had 4 sheep in the paper. But as we were kind of writing this this grant, we wanted to ask the question: well, how many sheep should we have used if we wanted to get more power into the experiment, into the experimental design? And so we were able to kind of again use our statistical test to kind of base our data on the observed variability from the experimental data that we had to estimate a group size that would be more powerful, and we kind of, you know, came to the conclusion that a group size of sort of 8 sheep would be better than a group size of 4 sheep. All right, and so that was really useful, right? Because we could then sort of put that into our research grant application and say, right, you know what, the next time we do this, we're going to have groups of a minimum of 8 sheep, and then that meant that we could have to use 12 sheep just to make sure that, you know, because obviously some might end up being lost to the experiment, but it allowed us to have a rational justification for how many animals we used, which was important. Unfortunately, we didn't get funding, but that's the way of it. We've all been there. So I just wanted to kind of finish things with something that is kind of another application that we have recently done with these data. So this is a research grant. So I'm just going to talk a little bit about some science that we've done. It's a research grant that that Jan was a co-I on, and actually Rupert, I can see your name is up here. So Rupert was on our advisory board as well. So and this was a research grant to look at antimicrobial resistance in the slurry tank of our dairy farm. And here is an image of the dairy farm that is at the University of Nottingham. If you ever come to the University of Nottingham's agricultural campus in Sutton Bonington, you'd always be most welcome to come and visit. And we have this lovely dairy farm here. It's actually bigger now. There's a new shed that is kind of well, on the other side of these old sheds. So we have a new dairy shed. It's even bigger. Many years ago, I went for. Ooh, I want to do that. That's no good. Many years ago, I went for a visit to the farm, and I got very interested in this structure here, which is the slurry tank of the dairy farm. And the reason why the slurry tank might appear to be interesting is because it contains a heady mix of bacteria, mainly from fecal material from associated with the cows, and antimicrobials of various kinds. They're cows, they get antibiotics primarily for mastitis, but they also get antibiotics for respiratory disease and for lameness. And particularly the antibiotics for mastitis end up in the slurry tank because the milk that has been produced by antibiotic treated cows doesn't then go into the main milk tank for sale, but is discarded into the tank. The cows also receive other kinds of antimicrobials, so antimicrobial foot washes, which at the time were copper and zinc, but and also some formaldehyde. Those things have changed since then, but that means that there's a range of antimicrobials in this tank, and we had a hypothesis that this seemed to be a selective environment for antimicrobial resistance. And of course, the slurry is then spread on land, onto fields and crop land as organic fertilizer, because it has value because of its nitrogen content. And so we were successful in getting some research funding to study this. Now, we did many things in this grant. Okay, one of the things we were kind of interested in is, how does the resistance content of the tank relate to altered patterns of farm antibiotic use? And then a really important question that came out, which was, can slurry storage help reduce AMR in slurry destined for land application? Now I put this question the other way around. So our original hypothesis was that we thought that the tank might be a place where we were selecting for resistance, but we've actually kind of come to the reverse conclusion that actually it might be a place that can help reduce AMR. And the key point that we were able to do with this is we could use modeling within this to test scenarios that would be unfeasible or unethical to perform empirically. But one thing I just wanted to show, there's a lot of data in this project, and one thing that I did want to show was samples from the tank where we have again used this DirtyGenes test. And so here we have metagenomics samples from the slurry tank taken at 5 different time points over the summer of what was 2017, so it was 5 years ago already. Time flies. And what I have plotted in these graphs are, on the left-hand side, the main bacterial taxonomic groups, and I have limited it to those that have at least 1% of the population. And the reason is that actually the DirtyGenes test tends to work better in terms of meeting the assumptions of the Dirichlet distribution hypothesis for those categories that have at least 1% of the population. And on the right hand side, I've done exactly the same with the antimicrobial resistance genes, or at least the gene classes. And what you can see here is that if you look at these data, is that they don't seem to change very much. And actually, this turns out to be a really important result. Okay, so the tank is kind of different things are happening. animals have been treated with different antibiotics, slurry is entering the tank. Obviously, there's whatever process is happening in the tank, so we think there's some quite significant changes happening in that population, but there's always fresh input from the cows. And in the end, we came up with a conclusion from our work, which is that actually the slurry tank is the material in the slurry tank is reasonably stable, because there's a dynamical equilibrium between what's entering from the cows, the fresh material, and then the processes that happen within the tank, the changes in the microbial community because of that environment. But one thing we were able to do again was to use the DirtyGenes test on both the bacterial taxonomic groups and on the resistance gene classes, in order to show that indeed the microbiology seems to be reasonably constant. And indeed, so we did this, and again, we got non-significant results. And in this case, the non-significant results were interesting for us because they were some of the evidence we had to show that these were things were not changing very much over time. We had other evidence as well. And I just wanted to show that because in the end, this is part of a much larger project, but we're able to use this test as part of the evidence to reach the conclusions we reached. So as I said, this statistic that I've developed has helped us to say, right, the microbiota appear to be in a state of dynamic equilibrium, and indeed these other results that we came to from this particular work that again you can read about in our paper if you wish, is that the resistance is maintained. It does actually reflect current and previous veterinary practice as well as interaction with broader environment, and it led us to conclude that the slurry tank is both a natural measurement point for on-farm resistance as well as a counter control measure point for resistance being released into the wider environment, and that's another story. So I just wanted to kind of thank all the people involved. Okay, so that's all I'm going to say in this presentation.
Thanks a lot, Dov. We've got plenty of time for questions. We can pick up other things as well that we haven't covered before. Please just feel free to put a question in the chat or comment in the chat. So I'm very happy, Dov, that you have time to present this today, because I think many people are not aware of the possibility of actually doing statistical tests on these data, because you don't see it used often.
Yeah. I should say that we do have the code on GitHub, but I should also say the code on GitHub needs updating and I haven't done it yet. So Jan, can I ask you a question then? I might have missed the beginning of the previous question because I did have to nip out briefly, but for your first question. But one of the things you talked about in terms of these oligotrophs is the fact that the counts increase as you dilute, which might also imply that you're getting a more, well, not the count, but the CFU per mil will increase as you get more dilute. The counts may actually go down because you're more dilute, but it might imply that there's a sort of a source of error associated just with the methodology itself. As you say, if your media is too concentrated, that will lead cells to kind of, potentially viable cells to die, and therefore there's a kind of an increased accuracy associated with diluting further, but then that's countered by the decreased accuracy of having a fewer counts. I'm just wondering how you kind of might take into account the fact that you might actually have better data for the reasons for the oligotrophs when you dilute further, but even though the counts go down, so your Poisson errors increase.
Yeah, I think I wasn't very clear about this dilution. So there are two very different things. One is the medium. If you dilute the medium, and that means rather than using LB in its normal strength, you use diluted LB, tenfold diluted, and actually this is something we have done in our grant in India. You get the larger number of colonies on the plate, and this can only be explained by the rich medium basically killing a lot of cells that are viable because they are able to grow on a medium that is closer to the environmental conditions, basically. So the medium is just not suitable.
So you're not talking about like just a tenfold dilution as part of the zero dilution series. You're talking about the actual medium itself being used in a more dilute form.
Yeah, but this is also possible. So you sometimes find that if you dilute more, you get a higher count than expected. And this can happen if there is a lot of competition, or if some of the bacteria in your sample are producing toxins like antibiotics that are inhibiting growth of the others. And then the dilution will allow others to grow in the absence of or reduce competition from the others. So that can happen. So it's always useful to look at your dilution series if you see unusual behavior, like a lot more colonies on the more diluted plate than you would expect from a 10-fold dilution, then it's probably some kind of competition or inhibition going on. If the opposite happens, it might be that the bacteria actually need other bacteria to grow. That's also possible. This is something that's, well, it's probably not very common, but there is always a possibility that bacteria only grow in the presence of others because they produce something that is required and is maybe not present in the medium, where it's just a signal compound. So there are some papers that have shown that if you're adding signaling compounds, things like quorum sensing signals, you can also get a higher count on your plates. So the idea that bacteria are pretty much autonomous and independent of any other bacteria, which is kind of underlying the idea of diluting samples, is not always correct. So this is a highly biased method in that sense. We will ignore any bacteria that require cooperation of some kind from others. Yeah, so the simple colony counts, if the more you think about it, the more complicated it really becomes what's happening. So just to get the discussion started on the DirtyGenes, so the software, is it written in R or is
it is in R, yes.
Yeah.
So there is an R script. I mean, we've had some good feedback. We've had a couple of people kind of send us stuff through, and we've updated the code on the Git. And then there was some changes I made for the when I did the analysis for the eval farms data, which I made the code to do that. And then I really need to get those reflected into the online version as well, which I haven't done yet.
So it's continuously being improved.
It is continuously being improved, still is, yes. And as I said, it's actually really nice when you get people contacting you to say, I think there may be a bug in your code. When I tried this, it didn't quite work properly. We're like, oh, right, that's great. Yes, you're right. And so you know, it is kind of, you know, it is a nice feeling when that happens.
Yeah.
And but there are a number, you know, because there's a number of things in there, so you can use it to you can actually, within the code, you can test whether or not the Dirichlet distribution is a good fit for your compositional data. So you can actually kind of run that statistical test on the goodness of fit of the Dirichlet distribution, and then you can sort of, you know, if that is rejected, then you know you don't want to use this methodology. It's not going to work. So that's actually quite a useful feature. As I said, you can use it for in a sort of power analysis mode, so you can actually do power analyses with it, which is something that we needed to do at the time. And then you can switch on, so you can make a large you can use it with a large sample estimation, so you can kind of switch off the randomization and just have it base the p-value on a chi-squared test. But in all the cases where we've done it, that would not be appropriate because the number of samples we've had have always been too small, and so we need to do a randomization test, and that then obviously takes a lot more time because you're generating, you know, 1,000 or 10,000 random samples to kind of do the test on. So we've done it on 3, 4, 5 libraries, right? So we can do it on really quite small numbers. If you have the randomization So that looks like 345. We know on 3 and 4 and 5, right? If you have the randomization on, that's fine, right? That's okay. If you want to use the chi-squared, that's not fine. You have to use sort of 20 or more. So it's a bit like the same sort of errors that Jan was talking about in terms of the Poisson distribution occur here as well. The other method that people use are methods based on something called Permanova, which is a non-parametric method. And the advantage of the Permanova methods is that you can use it in a slightly wider range of scenarios. So we've never extended our code to be able to deal with 2 or more factors in experimental design. It's perfectly possible to do so, but we just haven't. And obviously Lawrence has another job, so we just haven't done it. So there are advantages to using some of these other methods in that respect. And the main advantage of our method is, as I said, is you'll get the most power. So if the Dirichlet distribution fits the data, then you'll get the most amount of power out of the test. You'll get a more powerful test than Permanova. Obviously, if it doesn't, then what we're doing is not necessarily statistically meaningful. So I wouldn't recommend it. And then the other caveat of what we do is that it generally works best if you have anything you put into the statistical test as at least 1% of the population. So, for example, with the bacterial taxa in the Eval-Farms data, we had 175 different taxa in there, but we were only able to use about 7. So we could sort of stretch it to 0.5%, in which case you've got about 20 taxa. And it still worked, but I just decided, well, you get the same results. So I'm going to stick to the recommended 1%. It still makes the point we want to make, which is that the kind of the dominant taxa are not changing very much. And then the smaller taxa, it's harder to test. But you're going to have that problem, whatever statistic you're going to use, right? You're always going to have a problem with, again, if you've got sequencing data of a very large number of taxa in fairly small abundances, you're always going to have a problem of telling the difference between what might be changes and what might be random variation.
Yeah, that might be an important point more generally. So if you're dealing with metagenomic sequencing data, that if you have a large number of very small number of reads, or taxa with a small number of reads, or genes with a small number of reads, it's basically not possible to work out whether there are statistically significant differences in different samples, I guess.
Yeah, and I think that would be true of any method you use, right? You know, I don't think that would be, you know, I think it's a sort of a generic problem. So we just lump them all together as other, all right? But you know, but we appreciate again that actually there may be interesting things in there, right? It's not that it's not necessarily interesting. It's just that